And a very warm welcome to today's Cell and Gene Therapy Insights panel discussion, titled Entering the Fast Lane of Assay Validation for Cell and Gene Therapies. I'm Charlotte Barker, an editor at BioInsights, and joining me today are a panel of experts who, amongst other things, will discuss strategies for effective assay validation in the cell therapy space, with a particular focus on flow cytometry assays. As always, do feel free to pose your questions to our panel using the Ask a Question box at the bottom of the screen, and we'll try to get to them during the session. If you'd like to direct a question at a specific panelist, please indicate that in your message. I'd also like to draw your attention to the Resources tab on the right, where you can find more information on the topics covered today. So now I'd just like to briefly introduce our panel. Michelle Bruno is a seasoned expert in quality control within bioindustry. With extensive leadership experience, including roles as Head of Quality Control at Miltenyi Biotec and Director of Quality at Lentigen Technologies, Michelle brings invaluable insight into process optimization, regulatory compliance, and driving quality initiatives. Ajit Kamath is Senior Scientist 2 of Beam Therapeutics, working in analytical development. He has over 6 years of analytical development experience in developing phase-appropriate methods to support product development, including flow cytometry, immunophenotyping, and cell-based analytics. Christopher Bravery is Director of Consulting on Advanced Biologicals and Consulting Regulatory Scientist at Advanced Biologicals Limited. Advanced Biologicals Limited provides EU regulatory services to the regenerative medicine industry in addition to business and regulatory research and analysis. Christopher has a PhD in xenotransplantation immunology and spent 8 years in biotech before joining the MHRA as a quality CMC assessor in biologicals and biotechnology unit. Lily Wang is a NIST Fellow and leading expert in quantitative flow cytometry, critical diagnostics, and advanced therapeutic development. She serves as the Principal Manager for the NIST Flow Cytometry Standards Consortium, a major public-private partnership with key stakeholders from other agencies, industries, and academia, which addresses pressing measurements and standards needed for bioassays. So now, without further ado, let's get started with today's discussion. So I wanted to start with a bit of a scene-setting question. So finding out a little bit more about the different regulatory guidelines. So which regulatory guidelines should be considered in cell therapy assay validation? Christopher, perhaps you could start by giving us a quick overview of that.
Yes, well, there's really primarily just the main one that's international, which is ICH-Q2, but more recently there's been a life cycle version of that, Q14. And so actually the agencies have written rather few other guidelines for that, probably for that reason. But I did do my homework to make sure I could give you the exact titles. But in 2015, the FDA released a guideline, Analytical Procedures and Method Validation for Drugs and Biologics. And the United States Pharmacopoeia actually has a couple of chapters. It has the most relevant one would probably be 10.3.3, which is on biological assay validation. So anything above 1,000 in the USB is informational chapter. And then they have two which you must be wary of because they're intended for validating compendial methods. So that's intra and inter lab validation. But it's 12.25 and 12.10. So 12.25 is validation of compendial procedures, and 12.10 is a statistical guidance that goes with that. Beyond that, I mean, there's all sorts of other sources that could well be perfectly valid, and it's up to you to decide, which ones are the most useful to use. At the end of the day, it's scientific. So hopefully if you've followed any decent guideline, you've probably done a good job of validating and the regulator won't complain.
Michelle?
Sure. So I want to focus on two ICH guidelines, and thank you, Chris, for that good overview. I really wanted to look at ICH Q2 and Q12. So Q2, which is your validation of analytical procedures, and essentially that intent is to demonstrate fit for purpose. And, directly from the guidance, in practice, experimental work can be designed so that the appropriate performance characteristics are considered simultaneously to provide sound overall knowledge of the performance of the analytical procedure. And that's looking at specificity, selectivity, accuracy, precision over that, whatever the established reportable range is. Christopher also mentioned Q14. And if you, as you start looking at the regulatory guidances, you start seeing that they all interconnect and try to build on each other to provide a really nice framework to support your analytics throughout the life cycle of the product, whether it's early phase or late stage, right? Or commercial. So I think Q14, we start talking a little bit about system suitability, but it's a regular check of performance. But essentially, any sort of strategy that should be developed is based on the knowledge of the analytical procedure and the intended purpose. So it covers not only the intent of the assay, but it's important to remember that it needs to cover how the assay will be used routinely. It must be demonstrated that the assay is not only robust, but that it is sound under standard operating conditions. So, for example, the number of samples or replicates. And this is something that some people often forget. They just think, oh, I just need to do a standard number of replicates. But if you're not demonstrating how it actually works in your live operation, it's not gonna be robust enough, essentially. Additionally, at the end of this guideline, there are multiple tables that go through various types of assays and what should be considered for everything that you're looking at for validation. So that's specificity, precision, accuracy, reportable range, and/or robustness. So that's kind of a little snapshot of Q2. And then the other one that I did want to mention is Q12. So Q12 is your technical and regulatory considerations for pharmaceutical product lifecycle management, right? And so this is a great guidance that kind of gives you, again, that overarching broad view to help you really harness and provide an overview to manage the post-approval CMC changes. It ensures that it's done in a timely and efficient and predictable manner. And again, it links to other guidances and other guidelines. That's, ICH is all about harmonization, right? So they're gonna build and talk to each other. And it's intended to demonstrate how increased product knowledge can indicate future product needs. So it can tell you what you would need for your regulatory submissions, what the level of your reporting categories would be, and in general, just increase efficiencies. This guideline also links to important quality concepts like change management and knowledge management within the quality system. So just a quick note on change management, that's when you're looking at your system and you want to make a change. And this can be in your process, in your analytics, anything, even your facility build, right? So change management is a way that you control and you look at all of the players in the system. So you would look at your facilities, your engineers, then you would bring in your QC and you would bring in your QA and you would bring in your manufacturing and you do that risk assessment and say that incorporates ICHQ9 and you would incorporate and say, hey, how does this change really affect us, I should say? And what is the risk of doing this change? And what does this mean down the road? Right. So this kind of factors into that. So as you mature with your product and as your product as you gain more knowledge about your product, I should say, this is going to be really essential to have a really robust change management system. And that kind of ties into what Q12 really speaks to. So
absolutely. Ajit?
Yeah, thanks, Christopher, for that good overview, and thanks, Michelle, going into the Q2 and Q12 in detail. I can speak to a little bit more about Q14. So ICH Q14 actually complements ICH Q2 very well, and they actually go a little bit beyond and speak about fit for purpose method and phase appropriate method development, right? So in any assay development, we can't just develop the final version in the beginning. So it talks more about risk-based, scientific risk-based approach of developing your methods. And it all goes back to understanding what is the attribute of the product you're trying to measure with the assay and what is the target profile of the assay, what the assay should be measuring in what range, what's the precision required. For example, if you have a purity assay, right, you're going into very high ranges. So your precision may not be that important, but your range needs to be in that higher range. Whereas if you're looking at impurity assay, your lower limit of quantification and your precision needs to be really highly controlled in the beginning. So that's a very good way of thinking about what your assay is measuring, how you want to develop the final profile of the assay, and how you control it. And I think the second thing the Q14 talks a lot about is robustness. Like robustness is not something you do at the end. You start doing it in the beginning. You understand your assay very well. And in the beginning, you may not have practical knowledge, but you should have scientific information from your assay to understand what procedure parameters are critical for the assay and what are robust enough that you don't need to control at this point. So that's a really good way of like thinking about this. So, and I see Q14 actually has very good examples at the bottom to kind of guide the readers to understand, what like an example of like how assays look like and how you can control it in the different phases.
Great, thanks, Sajid. Lily, anything to add there?
Okay, is that fine? I want to thank, a previous three speakers. They really give, a pretty good view of the, guideline by the ICH. So I do see a lot of, cell and gene therapy producer, really, apply that to their basically product line. But however, I think, here I'd like to really mention about, CSI, that stands for, Clinic and Laboratory Standard Institute. They really focus on, really clinical lab, especially for that diagnostic purpose. So they have a lot of different, guidance document developed, under their organization. Certainly, NIST, FDA really contribute in a lot of their document development. So the particular one I'd like to mention is the H62. That really is the validation of assay performed by flow cytometry. So that's really relevant, as far as I can, tell through my entire career on quantitative flow cytometry. It's really the so one really details, how to validate, flow cytometry assay. So I guess, that document, including a lot of essential, basically chapters on instrument qualification, setup, standardization. That's, essential to, really have, basically warrant, assay, high quality assay. And that followed by assay development and optimization, analytical method validation, and then lastly, examination and the post examination phase. So that really covers, fit for purpose, using fit for purpose approach for validation of flow cytometry assay. So really, you need to consider, what's the, assay type and what's the, really the regulatory requirement, and also what is the risk factor for different type of assay. I think that's really good resources in terms of in assay steps, what proper control should be incorporated in terms of to really to have a high quality assay developed, and how in terms of the intended use of the assay, how you should develop a basically a strategy for validating those assays. So that's covered really in that document.
Can I ask Lily a question on that, actually? Because obviously that's intended for clinical use of flow cytometry. Do you see any particular differences in the needs in the way that you would go about validating for a clinical diagnostic versus manufacturing?
Yes, I certainly, feel, like those, basically validation strategies certainly apply because, flow assays, multi-step process. So in terms of diagnostic, in terms of, also, biomanufacturing and the cell and gene therapy, that's the same flow assay. I think that's certainly, right now, because of cell and gene therapy now is more doing, approaching, to in vivo, by engineering. Certainly now is making diagnostic and the biomanufacturing, the differences are, drastically minimized. So I'm guessing, there's, very little, I mean, differences between the two categories, in my, opinion. And also, for example, a lot of assay, how to, qualify the assay in terms of, the antibody, how you, evaluate the antibody and how you really perform compensation. All those things, I think, is, apply not only to diagnostic assay, but also apply to, cell characterization for your, therapy product, right?
Perfect. Thank you, Lily. So what are the main challenges in validating cell therapy assays, particularly with flow cytometry? And Lily, perhaps you could start us off there.
Yes, and this, since I'm serving as committee member during the H62 document, development. So certainly, doing that, document writing, we really realize, there's a huge, in terms of challenging for, basically high quality flow cytometry assay. So particularly, since, there are a lot of, parameters, in order to validate the assay, including selectivity, precision, a lot of different, parameter. But however, the accuracy and trueness and also linearity, are the two biggest challenges because of a lack of, high quality reference material. So allow you to, basically to derive, calibration curve, and then your unknown can be, really, characterized in terms of against the calibration curve. But that certainly is, we feel is the biggest challenge in develop a high, high quality flow cytometry assay, and certainly, validating those assay. I think, so far, I think, through flow cytometry standard consortium, led by NIST, we, deal very well in terms of instrument standardization using NIST, equivalent number of reference flow for calibration service. So we assign fluorescence unit to calibration material, basically, provided to NIST for assignment. So that enable, instrument, calibration and also standardization using the, ERF concept that is traceable to high order, high quality reference flow for standard, produced by NIST. But however, in terms of a biological, side, the lack of, really biological relevant high order, reference standard, that's the, the bottleneck in terms of, for, clinical diagnostic, certainly for the, cell and gene therapy application field.
Thanks, Lily. Ajit?
Yeah, I mean, like the instruments are really important, but one more challenge that we like always think about is operators for flow assays. Like still flow assays have a subjective nature to them. Like, how you gate, where do you draw your lines? How do you make sure this is the right populations? Controls obviously help there, but biological samples are very variable. So operator variability is something that we really have like a big challenge, and we're always working toward like how to best characterize like that and how to control that variability in the validations.
Christopher?
Yeah, I think I'll broaden it out a little bit beyond flow cytometry as well, because I think there are quite a lot of challenges for validation. And I think the comment that Lily made about the lack of reference materials is fundamentally a problem for all sorts of the assays that we do, even as fundamentally straightforward as counting cells. Where's that 100% viable supply of your cells, your exact cells, or cells that are near enough to yours to be relevant, versus the 100% dead ones? Well, they don't exist. So you have to play around with things like, well, if I do serial dilutions, then I expect to see serially reduced amounts of cells and sort of deduce accuracy that way. I mean, in some ways, compared to clinical measurements by flow cytometry or other methodologies, sometimes accuracy is not necessarily that important in manufacturing. It's precision that matters. you want to know that every time you make that measurement, you'll get the same result, even if that result is wrong. So, because fundamentally, you just want to keep your quality attributes in the same place. So I guess we have that benefit over perhaps other forms of measurement where accuracy might actually be really important. But of course, if we're measuring an impurity and we know that that's dangerous, then accuracy will be important. And I think that's where we enter some serious challenges in some realms of cell therapy. For instance, if you're using embryonic stem cells and you want to make sure that there aren't any in your product, that they've all been differentiated or lost. And I think flow cytometry is a tough job once you get below about 1%. Being sure that you're measuring something accurately is difficult, and I'd be interested in anybody who got any words of wisdom about how to do that. The final thing I want to say is just to go back actually to reference materials. One of the areas that I have a particular interest in is potency assays. And flow cytometry is often used for potency assays. But the bit that tends to be overlooked is when you use things like flow cytometry, or maybe you use an ELISA, or maybe you know, etc. Those are the readouts of your potency assay. So when it comes to validating, it's easy to validate the readout. It's the underlying bioassay that's the problem. And in cell therapy, having a product reference standard is highly problematic because viable cells are the antithesis of what you'd want as a reference material. They are entirely unstable. Look at them and they change. You know, look at them through a door when they can't see you looking at them, and they'll change. So we want something very stable for a reference standard. So that's also causes us a big challenge, and I'm sure we'll come back to that point in another question as well.
Can I comment? I think, you know, I really appreciate Christopher's, you know, comments. So I guess I want to also mention, you know, ISO does have, you know, produced, you know, cell counting, you know, documentary, you know, guidance. So certainly that, you know, needs to contribute in terms of write-up. So that, you know, I think, you know, advocate, you know, severe dilution of the, you know, basically cell sample in order to really, you know, derive, you know, accuracy of the cell count. You know, I think the other, you know, certainly FDA released, you know, guidance, you know, document for consideration of for the development of CAR T therapy. I think, you know, January of this year really, you know, specifies, you know, flow cytometry as a critical analytical tool for a lot of, you know, critical quality attribute of the cell and gene therapy, you know, product, including, you know, viability, you know, cell count, purity, you know, certainly identity and the strengths. That also echoes to Christopher's, you know, comment about, you know, how you evaluate the potency. Certainly, you know, potency, you know, for therapy, there are different way to evaluate through the, you know, the therapy manufacturing, and also, you know, at a different stage. You know, you can using ELISA, you can also using, you know, flow, and you know, also doing, you know, basically identify, you know, critical component, you know, central memory phenotype in the, you know, viable cells for sure, and also cell killing assay, you know, in vitro cell killing assay. So all those things, I think it does requires, you know, validation of those assays, you know, during the product development, eventually, you know, goes to clinic. I think, you know, that's also posed, you know, huge, you know, challenging for, you know, especially flow play critical role, how to validate in different, you know, I think, you know, application, you know, scenario. And certainly, I hope, you know, everyone, you know, aware, you know, FDA's, you know, really recommendation in terms of fit for purpose, you know, risk assessment. So that certainly poses a lot of challenge in front of us. That's also, next flow cytometry standard consortium for the next phase of, really, investigation in terms of developer, cell therapy, really, mimic, or reference material to enable assay validation. So, we really, appreciate the work with the consortium member and also certainly, Miltenyi to produce, really engineered, T cell, product for next phase interlab study. So all the consortium member to, really can evaluate using, really common standard operating procedure to characterizing those cell product that essentially not only really generate a best practice procedure, but also for the future likely reference material available for manufacturers to really benchmarking their therapy product.
Thanks, Ali. Michelle?
Yeah, I think the main thing that I would I think my esteemed panel has really covered many different topics here, but I think the main thing that I think I would think about just in terms of some challenges would be, once we start characterizing early on, and this is specific to flow cytometry, that we might have a really big panel, right? Or a large number of colors. And that is going to be very hard to basically to validate, to qualify. And we might release on a smaller number of colors or panels, right? But then as we go along, we realize that we need to characterize more fully. So that time that can change your approach to how you qualified versus how you validated. And you kind of have to do it piecemeal and really be pragmatic and strategic in how you do that. So that can be a really big challenge. Just something I wanted to quickly add.
Can I quickly follow up on that? Because regulators have always been quite clear on this. Any tests that you're using for releasing a product have to be fully validated eventually. I'm sure we'll get into the topic of when and where. But other methods that you might use for extended characterization, they only need to be qualified. And that's a very vague term that they do not define what they mean by. It's up to you to decide it. So the benefit there is when you do get to your many colors, this is not something that has to be necessarily fully validated to an ICHQ2 type standard. It just needs to be sufficiently reliable for whatever purpose you're going to put it to. For example, the process changes that were mentioned in relation to Q12, for example, that would be a situation where you were supplementing your comparability data with a qualified method result on top of your validated release tests.
Brilliant. Well, to follow up on that, which assays do need validating and when, and also can accelerated development timeframes have an impact on that? Ajit.
Yeah, no, thanks, Christopher. Like that actually does lead into this question, right? Like, release assays need full validation and your characterization need qualification. And again, that comes back to Q14, right? You need a risk-based approach to know what qualification means for your assay, right? You need to understand if you need precision, just do precision. If you need range, do that, right? But in terms of I can speak to a little bit more about development timelines and timing. It all comes back to you need to identify what are the critical attributes of your product early on, and you understand what assay is targeting which attribute. And broadly, you want to break down attributes into two main things. Does it impact the safety of your product or efficacy? If it's safety, do that first, right? That needs validation sooner. You need to get your assays in line and make sure it's working well first, and efficacy comes a bit later. So it gives you a little bit more flexibility there. But we all know the timelines today are really breakneck speed, right? So when you're developing assays, I would you always tell people make sure you understand your assay early on when you're developing them. You identify what your critical assay parameters are and start bringing them under control sooner rather than later, right? You don't need them under control till another 6 months, but if you can do it now, let's just do it now. So yeah, again, coming back to the same point, safety, efficacy, if you can break it down that way, that always helps to speed it up.
Definitely. Anything to add there, Lily?
Yes, I think certainly, when you try to validate the assay, that's really, you have to really, have an idea what is the assay intended to use, and certainly what the regulatory requirement. So I think those are the two critical components. But also, you have to really be aware, the assay type. I think, as, Ajit say, during the development phase and all the way to the manufacturer and to the, apply to patient. So they are, different, certainly risk factor you have to really consider. I think, in terms of those things, I think certainly, risk assessment, I think, really COSI H62 give, many example in terms of the type of assay and also the, the category of data and also risk category, and try to fulfill, regulatory, requirement. So I think, those, it will serve a good, template, although it's not a, strict, basically you have to validate the assay, using, specific, type of a sample and also, replicates. But I think it provided a, minimal requirement, for users to consider, about validating the, assay. So I guess, certainly, assay type, would be, pre, development or development and certainly all the way to, clinical, utilization. So I think, certainly I want users really, based on all the ICH, guidance document, provide a big frame, but also was more detailed in terms of how you validate assay using, basically according to, H62, recommendation. I think that certainly helps a lot in terms of, validation strategy and how to, really plan out, validation phase.
Lily, I'm going to tack right back onto your word strategy and look at it from a slightly different point of view. I think that we are all trying to go through our development phases as quickly as we can. And I just always like to say that when we get to a point where we're saying, okay, we're talking about validation, that we have ensured that we developed a strategy along the way that, like what Ajit had spoken to, that robustness was done early in the development, right? That we have fully characterized and understood our analytic target profile, right? Q14, that we have understood exactly what we are trying to look at with our analytics, and that we have enough data from our qualification to really do, or our pre-validation work. I think I like to use pre-validation instead of qualification, that we have that data there to really do a substantial validation, because once you move into validation, you start adding on quality implications, right? In terms of change control that I mentioned, in terms of, just notifications, regulatory submissions, filings, things like that. Once an assay is validated, there's a different level of scrutiny that is placed on that assay. So just to make sure the strategy is there when you're thinking about when you're going to validate, when you're going to do that, that you factor in those timelines and that strategy to make sure that you are truly ready to validate, and that you have a strong assay to validate, right? That you've done the work up front and that you've done that. So
Thank you, Michelle. Christopher?
Well, I'm strangely going to step out of my regulatory mode a little bit and go into more commercial mode and just say, well, pragmatically as well, from a development perspective, if you say literally validated fully all your methods before you've even started, this would be a huge drain on resources. And you might not even get through your first in human. You might actually just, fail the program at that point. Admittedly, not so many cell therapies get failed probably in first in human as, say, small molecules, where rather a lot do. So this idea that you would fully validate, is a little bit nonsensical anyway, because it would just be too much work, too expensive, and we'd never get off the ground. But I just warn the listeners out there that this term validation and qualification, I battle with it constantly with all the people I work with. I had a conversation with a contract manufacturer recently, and I said, no, we just need to qualify this as a first in human. And they said, well, we don't use that term. And apparently they used the term validation. And I said to them, that's not very helpful, is it? It's much more useful to have a different term to validation, so we know that we're talking about something different. And if you call it all validation, is actually not very helpful. So you do have to be very wary whenever you have a conversation that you're even talking about the same thing. You know, some people use validation with a little v and validation with a big V and all sorts of weird things to try and make these distinctions. So what do we mean? Well, actually, it would be quite nice if we had something more prescriptive, but in a way, it's also much more flexible. So I think, you know, I think what you've all said is I agree with. You need to focus on what's important about this assay, how important is this assay, and do a commensurate amount of work to make sure that you feel comfortable that this assay is reliable enough.
Yeah, so once we've decided we definitely are all talking about validation, let's dig into that a bit more. What is essential in designing assay validation strategies? And how do QTPP and ATP inform analytical procedure development? Lily?
Oh, I think I covered this one a little bit earlier, but I still feel, emphasize, the context of use. Certainly you have to make sure, what is the assay used for, and certainly what's the, regulatory requirement. So those are the two, critical, things, basically, user have to really think thoroughly through. I think also, as Michelle said, really at the different phase, you really, I think I like Michelle's, saying in terms of qualification, assay qualification, and certainly that's. To us, that's a little bit little work, and it can be done in the resource confined stage, because doing the product development for cell and gene therapy, you have many phases, but you always every phases you have your own assays, But those assay, certainly as I just say, you should really know what is the assay intended for. But also certainly the assay has to be qualified at the beginning. So I'm guessing that's the stage we think you have a multiple assay at a different stage. But however, eventually, really for releasing assay, that will depend on the assay. What is the assay mostly really impact for characterizing your cellular product for supporting its submission. So certainly that assay then developer have to validate that assay. I think it's really the one you have to provide the data quality and also risk assessment, all those validation parameter in terms of I think selectivity, precision. As Christopher say, precision is critical, but I think it's also more and more so for example in terms of CAR T therapy space, you have to really able to detect the CAR and detect the expression of the CAR, so that I think is really up to quantification assay, you quantify the CAR expression. So that I think so far, because of lacking of reference standard, that it really is challenging in terms of full characterization or validation of the assay. So I that's my some comments in that category.
So if we look at our basically our QTPPs, we are looking at our quality target profile. I am obviously as a quality personnel, like this one always had stood out to me. And I'll take a little bit of time to like kind of go into that, but it's sufficient to, to understand your quality target profile, ensure that you're basically developing your method and your assay in the direction to ensure that appropriate tests are being performed, the method's fit for use, the method's ability to provide accurate information regarding the product specificity, accuracy, precision, and reportable range. Note that this kind of sounds like qualification, right? And if you look at your analytical target profile, again, that's pulling from Q14, ICH Q14, and specifically it speaks to that consisting of a description of the intended purpose of, the analytical procedure, right? So that you would have appropriate details on the product attributes, what's going to be measured, and relevant performance characteristics and performance criteria. So your ATP is going to inform the choice of the appropriate analytical instrumentation as well, right? So you've got your ATP and then your QTP, really working together. And you can use that so that you can understand and track your changes and consistently check in with your current QTPP and your ATP as your product knowledge matures, right? So if you understand your target profile and you're understanding your analytical target profile, you can use that. And as your product matures, you can use that as a gating function to understand your product knowledge. And so your data trending is critical here. You have to have sufficient data trending and establishment of that profile to ensure that you are really working to understand your product maturity. One thing that I'll note here is that it's hard in the cell and gene therapy space sometimes to understand that is that when you look at your healthy donors versus your patient profiles, that might be a little hard to synergize that and to understand that as you're developing and looking more towards a personalized form of delivery, right? So that's always been the crux within the cell and gene therapy space. But as far as QTPs and ATPs, that's kind of where, I would go. I think it's critical for product maturation. And those two things are really strong tools in your toolbox, I should say, to help understand your product development.
So just say, I wish that the myriad of cell therapy developers I worked with over the years had the resources to actually do what you just described. And it comes back to when do you qualify and when do you validate, et cetera, et cetera. Mostly they start thinking about things like QTPPs very much later in development because they just don't have the resources. But, that is changing with more money coming in and bigger companies. Actually, I just wanted to throw in something I forgot to mention in the problems section, and I realized that, well, maybe I should just say it now, otherwise I'll totally forget it. This is a little warning to all of you out there, actually. I refer to this as the kitification of science. So, I'm old enough in the tooth that, when I used to do an ELISA or something, I'd design it from scratch when I was in research lab. Yeah. I'd get some antibodies and I'd make my own ELISA. These days, you just go and buy a kit from somewhere. Likewise, if you're going to do cell counting, you buy a nice little kit in a machine. And if you're going to measure cytokines, you might buy some fancy multiplex gizmo that, you just put a sample in, it gives you loads of values, just like that. Now, my warning to you is that this kitification of science means that sometimes the analyst does not really understand what the machine is doing. And then when it comes to validating, mistakes get made. And I have spotted a few recently in qualification efforts of my clients, and it really did come back to them not quite understanding the device. And it also happens when you move from one method to another. I mean, one anecdote I'll give you was for a potency assay that was based on measuring a cytokine release. I won't say any more than that, but it's quite a common approach. The company had been using ELISA methodology type machine 1, And then someone said, oh, there's a nice all singing, all dancing, much sexier version of this machine by another company. And it does it much more quickly, smaller sample, whatever, I don't know. So they wanted to change their technology. Guess what? Machine 2 gave them a completely different result. And I asked, well, what reference standard did you use then? What calibrator? Well, the one that came with the kit. Okay. Did you consider swapping the ones between the kits? Oh, no, we didn't think of that. Well, you might, therein may lie the problem. So, yeah, so it's worth, you know, thinking about some of these things up front, making sure you really do understand how your machine works.
Yeah, I want to concur, you know, Christopher's, you know, the last comments. So that's also, you know, we experience a lot, you know, in terms of, you know, how, you know, really the commercial kits really, you know, works. You know, I think a user have to really, you know, understand it, you know, basically, you know, since, you know, kids from one vendor and the kid from other vendor, they using their, you know, working standard, basically manufactured by the, you know, the producer, you know, certainly not with the same, you know, basically property in terms of, you know, I think a specificity to whatever platform you use. So that's also the, you know, the things that you consider is, you know, our approach in terms of, you know, standard consultation, you know, really, you know, identify, basically separate the instrument, you know, issue from biological. So that's, I think, you know, is broadly, you know, applicable to, you know, all the different, you know, assay platform, including ELISA. But also, yeah, I think for user to keep a, you know, eye on it is, you know, not just to say, you know, I'm using one particular vendor's, you know, kit, you know, to perform the assay validation, but, you know, certainly kick in terms of high order reference material, certainly, you know, available. For example, you know, the cytokine, certainly, you know, soluble cytokine manufactured by, you know, I think, you know, in IBSC, you know, so it's a WHO, you know, designate the blood institution. So they produce, you know, cytokine reference, you know, standard. So those things, I think, for user, you know, really should consider, you know, utilize whenever the high order reference standard are available.
Sorry,
so
I'm gonna have to intercede with that because you might be misleading the audience slightly. Yes, they have cytokine reference standards, but they're for biological activity. They are not for milligrams, and that's a really important distinction. So they are designed for activity, for measurement in international units, not for calibrating. You know, how many milligrams per mil did I find in the sample?
That's correct.
Yeah, and I say that because there's a couple of very interesting papers, and I was thinking maybe I should have provided them as a reading material or something if I could find them again, where they'd looked at these sort of multiplex systems and taken something like 4 different suppliers, and looked to see how they differed in the cytokine results that they measured. And it was both these papers were quite shocking, really, how different the results she got with the different machines were. And one of the ways they tried to standardize that was using the WHO reference standards. And I found myself shouting at the paper, you should not have done that. That's naughty. That's not what they're there for. They're for activity, not for mass.
That's true. I think, you know, I think you're right. So what I mean, I'm trying to give example. If there is, you know, for example, standard available, certainly should apply. But, you know, I think certainly, you know, in terms of, you know, shift from one instrument platform to another, that's, you know, kind of, you know, approach done by flow cytometer user because of advancement of instrumentation. So really the user really have a lot of issue in terms of how to translate their the clearance assay to different instrument platform. So, you know, so that for that, you know, basically the issue. So certainly, you know, should using strategy like a separate instrument, you know, performance from biological, you know, variability. So that's the one I will try to, you know, basically emphasize through the comment.
So how do you tailor assay validation methods to cell therapy characteristics? And how can flow cytometry adapt to these requirements? Ajit.
Yeah, I think we spoke a lot about this earlier as well today, but it comes back to, identifying attributes of a product and what attribute this assay is measuring. I think one thing that we like briefly touched upon, I kind of want to elaborate here, is the sample type. With most cell therapy products, we use healthy donor material to like develop assays, do the pre-qualification, early robustness. But one thing people we want to understand is when you have true patient samples, those matrix effects can wreak havoc with your assay, and you want to be really cognizant of this. And many of the data will be available only after you have a few clinical lots under your belt. So in the beginning, I would like be very cautious about locking your method. Make sure you have flexible gating, you have flexible some sample prep steps, like example, lysis of lysis reagents, your buffers. Uh, but let's try to like gather data on that, and that helps you generate more information on your assay procedure parameters that you can start validating overall. One of the interesting things which people talked about was compensation. That's something we also want to think about in terms of like in context of the patient material, right? Because sometimes you can have weird compensation effects that show up with your actual patient material and don't show up in healthy donors. So if your stuff doesn't look the same, I would also like go back to compensation and see if we can improve that going forward. And finally, like, trying to validate your method. You have all your procedure parameters and whatnot. We want to really think of your compensation and your instruments as part of the method, and they are not a separate thing, because your instrumentation, your lasers, your Levi Jennings, they do impact how your assay looks like. So I would really start thinking about how to incorporate them in also as part of your assay workflow, rather than just having it as like its own thing.
Yeah, I concur what Ajit said, but I do have a comments in terms of compensation. I think compensation is really the instrument specific task. I think it doesn't depend on the sample type. Certainly, once you compensate the instrument, and certainly through Miltenyi plot, you can monitor compensation stability. As long as your instrument, qualification is consistent through time period. So, but another, consideration certainly, as Ajit say, different patient, certainly, patient sample is, very challenging because of different disease state, and also a lot of time patient through, different, therapy regimen, and really post a lot of issue in terms of really the true measurement. So certainly the compensation may need to take a close look at to see whether it's still applicable to specific patient sample. But I do feel compensation is instrument dependent task, should not depend on the sample type. But however, keep eye on certainly in real case, keep eye on the patient because of complexity of patient sample.
I mean, we have mostly been talking about using these methods and validating methods that are used for manufacturing, but I see we've drifted into the clinical, and indeed we might as well throw preclinical or non-clinical studies into this discussion at this point as well. Then clearly the matrix is going to be very, very different, as you were saying, and indeed. Usually they're done by different people, actually. So they do tend to be separately validated. And when you think of that difference, in manufacturing, when you're, I don't know, making a CAR T, then you've got 90+% CD3 cells, at least maybe 50% of which are modified. When you take a patient sample, you're looking for a much rarer event. Possibly with lots of B cells around, or possibly not, I don't know, as you say. So I think the challenges have become very, very different. So almost certainly it's different people validating separately those methods, actually. Although, they should learn from each other, I'm sure. So Other than that, I'm not sure I've got anything more to add to this particular topic.
Can like elaborate a bit more, right? Like when I meant matrix, it was actually matrix of the patient cells in manufacturing. Like sometimes you can have certain markers expressed on diseased patients actually which are not visible in your healthy donors, and your T cell characteristics can be impacted.
Oh, you were talking about the starting material.
Yeah, and
Yes,
it
yes.
actually
I thought
comes back
you
into
drifted into clinical testing. Sorry. Yes, yes, and in fact, Lily, I think already made the comment about the difficulty of how we need to sometimes do process development with healthy donors, and versus the product being made from patients, and that does cause all sorts of difficulties. And somewhere along the line, you've got to justify that it is relevant and valid. I have seen people try and sort of concoct patient-looking samples by fiddling with it. I never felt very comfortable with that. I wasn't sure that was a good solution. But you know, every product's different, so it's up to you, I guess, to justify what you do. But yes.
I have a last comments. I think because of the recently approved FDA guidance guideline, so certainly also recommended cell and gene manufacturer also really have to monitor patient for additional 15 years after therapy administration, so that also make the clinic and also cell and gene manufacturing, the difference is almost none. You have to monitor, so that I think certainly post also challenging the flow cytometry assay validation task, because you have to consider both, and not only study material from patient but also monitor the therapy efficacy for 15 years. So that I think it is the future. So that's the critical topic you try to cover. Thank you.
So which innovative technologies can enable flow cytometry assay validation, and how can flow cytometry workflows enable regulatory compliance? Michelle.
Sure, thank you. And I'm going to actually bring it in internally to some really great things that we have here at Miltenyi Biotec. So we rely on what's called the workflow solution, and it offers several features and enhances our validation process. So we have recombinantly engineered antibodies that ensure consistent performance across various batches. We have extended validation data from this, that ensures both high specificity and sensitivity. And there's something called Smart Gain technology, which is in the MaxQuant analyzers that transfer settings between instruments. And I know, Lily, you were speaking to this before, and it becomes very effortless and ensures consistency across the different sites. The software is also critically important here. So within the software, we have automated gating algorithms that are essential to streamlining data analysis and eliminating that human bias. And the Levi Jennings and daily QC functionalities maintain a high assay quality. And then finally, the Max Quantify, or the Max Quantify, excuse me, software can be upgraded to a 21 CFR Part 11 module, which allows us to refine process documentation, manage the user roles, and ensure compliance with ALCOA principles for data integrity, and which is obviously crucial for regulatory submissions. So that really helps us to align nicely with regulatory needs. Finally, the Miltenyi workflow supports assay validation for cell and gene therapies through integration of the MaxQuant into automation systems, and that facilitates scalability. So furthermore, the automated compensation feature on the MaxQuant ensures instrument settings are updated with daily calibrations. They enhance the accuracy and reliability of our assay validation process. So altogether, these features integrate seamlessly to support a smooth and robust validation process. So that's kind of my little elevator speech around our flow cytometry assay validation and how we help to enable it with our MaxQuant instrumentation and software that we have here at Miltenyi Biotec.
One of the main things for flow is identifying what is your raw data and what's your process data, and trying to have a good way of documenting them into your systems, like whatever that system will be, right? Because flow, you have the raw data, which can always be manipulated later. They can always process it different ways. So it's really important to have that link. And a lot of 21 CFR softwares try to do that very well, right? Like, so that's one of the things I would say that we really want to be careful of. And that actually comes back to ATP. When you're choosing an instrument, make sure you try to answer this question early on, which is very difficult to change instruments once your assay is like deployed already.
Yeah, I would just warn people that to remember that with particularly something like flow cytometry, you've got a lot of what we would call critical and analytical reagents. So all your antibodies and possibly some of the other reagents that you've got. And that means slightly less during development, but it still means that, you know, you can't just swap that for somebody else's antibody without requalifying or revalidating. And when you're post-approval, that means any changes to those analytical reagents would require notification of the regulator and approval before you could implement. So they have to be handled with quite carefully.
Yeah, I concur, you know, basically what Christopher say, and I think, you know, certainly Michelle really, you know, give, you know, the strategy, how they handle in terms of the, you know, basically streamline the process to really enable, you know, relative, you know, easy, you know, basically, you know, approval, you know, so streamline those processes. But however, I think as Christopher say, you know, all those reagent, you know, certainly, you know, any change you have to revalidate your assay. So that's also, you know, means, importance of, control material, for example, cell reference control, so we, working with, a lot of consortium member try to basically come up with a solution in terms of, user can using, reference material to validate their antibody. So everyone using the same, certainly you can, really, serve as a validation tool, enable that. So I, not only that, I think also, during the H62, development, we realize, for example, they are not in public domain, really have data backed up in terms of, what is the compensation matrix, quality, for example, cell-based or, bead-based. So certainly, that, it's not a really systematic evaluation in terms of those in the public domain. So that's also really, posed a lot of, really, measurement issue in terms of, development of, assay and optimizing the assay even prior to, validate the assay. So all those things, I think, we certainly really welcome, audience, try to take a look at, our approach undertaken by the, flow cytometry standard consortium. So we try to, really, build up a solid foundation in terms of measurement process and also, reference control material to bench up, for the, easy up for, development the assay and certainly, help to, assay validation process.
Brilliant. Well, thank you very much, Michelle, Ajit, Christopher and Lily, for an informative and thought-provoking discussion. That is all we've got time for today. Any questions that we didn't get to, we'll reply by email. Today's webinar will be available on demand tomorrow, so look out for an email from us with the link. And I think all that's left then is to thank our panel once more for a great discussion, and thank you to the audience for listening. We do hope you'll join us again soon.